Once again, I welcome you all to the biology classroom. We are discussing our second chapter, Reproduction in Flowering Plants. In the last class, I just gave you an introduction about the reproduction in flowering plants. Today, we are going to discuss about microsporogenesis. So, what is microsporogenesis? The formation of pollen grain from pollen mother cell, that is PMC, is known as microsporogenesis. When the anther develops, the cells of sporogenous tissue undergoes meiotic division to form microspore tetrad. Microspores are usually arranged as 4 cells, hence called microspore tetrad. So, the microspore mother cell undergoes meiosis division 1 and 2 and it forms microspore tetrad. From this microspore tetrad, each microspore develops into an anther. I think it is clear from this picture how pollen grains are formed from microspore mother cell. Pollen grain represent the male gametophyte. When we observe the pollen grain through a microscope, we can see variety of architectural size, shape, color, etc. Pollen grains are generally spherical, measuring about 25 to 50 micrometer. It has a prominent two-layered wall, the outer exine, which is made up of sporopollenin. Sporopollenin is one of the most resistant organic material which can withstand high temperature and strong acid and alkali. No enzyme can degrade sporopollenin. Pollen grain exine has prominent aperture called the germ pore. It is because of this sporopollenin that pollen grains are preserved as fossil. The inner wall of the pollen grain is the intine. Intine is thin, continuous layer made up of cellulose and pectin. The cytoplasm of the pollen grain is surrounded by plasma membrane. When pollen grain matures, it has two cells, vegetative cell and generative cell. Vegetative cell is bigger with abundant food reserve and irregularly shaped nucleus. And generative cell is small, which floats in cytoplasm of vegetative cell. Generative cell is spindle shaped with dense cytoplasm and a nucleus. This stage is called the two-cell stage of pollen grain. About 60% of angiosperm pollen grains are shed at this stage. The generative cell divides to two male gametes. This stage is called the three-cell stage. In three-cell stage, in pollen grain, they have three cells. One is a vegetative cell and the other two are the two male gametes. So altogether, they have 3 cells at this stage. Pollen grain may cause disease like allergies and bronchial affliction, which may lead to chronic respiratory disorders. Parthentum, which is known as carrot grass, is a common plant which comes to India from imported wheat, causes pollen allergy. Nowadays, pollen grains also have market value because it is highly nutritious. Pollen tablet and syrup are available in the market of Western countries. It claims the better performance of athletes and race horses. After dehiscence, the pollen grains have to reach the female reproductive part of the plant for fertilization. All these things must happen before it loses its viability. The duration of viability of pollen grain varies in accordance to plants. Some plants' pollen grains viability is about 30 minutes, and some plants like wheat, family Rosaceae, Leguminosae, etc. have 1 month viability. So what is viability is the ability of the pollen grain to perform its action as a viability. So losing viability means the pollen grain loses its ability of fertilization. We know about storing sperms of animals, including human beings, for artificial insemination. Like this, we can store pollen grains of certain large number of species for years in liquid nitrogen. This is called the pollen bank. This is up to today's class. Hope you all understood what we studied in this video. Thank you. Have a nice day.